I'm pleased to introduce Dr. Jeannie Chin. She's an associate professor of neuroscience. Her research uses a multidisciplinary approach to better understand the cellular and network mechanisms underlying Alzheimer's disease and seizures. Her work points to the long-lasting changes following even low-frequency seizures conferred by epigenetics, insights that may serve to identify potential therapeutic strategies for the treatment of these devastating diseases. Congratulations to Dr. Chin. Please watch this video for more information on her recently published work.
Well, thank you so much for giving me this opportunity to tell you a little bit about the research that my lab has been doing over the last several years. I'm going to be talking about what we've discovered about mechanisms of seizure-induced cognitive deficits in Alzheimer's disease. My lab has had a long-standing interest in understanding the mechanisms underlying cognitive impairments in neurological diseases, including Alzheimer's disease. For example, what exactly are the cellular, molecular, and network mechanisms by which Alzheimer's disease leads to learning and memory deficits? Most people are familiar with the amyloid plaques and neurofibrillary tangles that are hallmarks of the disease. However, these are relatively late stage, and it turns out that Alzheimer's disease is way more than just plaques and tangles. There are many subtle effects that are very important to the development and progression of the disease, and that's what my lab is very interested in understanding. We've learned a lot about Alzheimer's disease from familial autosomal dominant forms of Alzheimer's disease that include families that have mutations in presenilin, one of the secretases that cleaves APP, and also for mutations in APP or amyloid precursor protein, or even duplications of the amyloid precursor protein gene. All of these autosomal dominant mutations lead to Alzheimer's disease. And so what they've done, or what people have understood, is that all of these mutations lead to an increase in the overall levels of the amyloid beta or A-beta peptide. And so much research has really focused on trying to understand what is A-beta's contribution to the learning and memory deficits and other behavioral impairments in Alzheimer's disease. Many transgenic mouse models of the disease have been created that mimic these mutations. So mice with presenilin mutations, APP mutations, or overexpression of APP have been developed to really understand what is going on in Alzheimer's disease. And just like the patients, these mice also produce high levels of A-beta and develop learning and memory deficits. And so we use one of these mouse lines called the J20 line. You can see if I look at just disease progression over the first 6 months of life in these mice, they start to produce A-beta very early on. You can see it start to accumulating reliably by 1 month of age or so, synaptic loss by 2.5 months of age or so, cognitive deficits by 3 months of age, and they don't form plaques or deposit plaques until about 6 months of age. So in my lab, we're very interested in trying to understand what happens in this early stage of Alzheimer's disease progression in these mice. How does A-beta lead to cognitive deficits? And is there a biomarker of early events that we can either target early in disease or use to help diagnose Alzheimer's disease earlier in disease progression? And two of the phenotypes that my lab has really concentrated on in the last 5 years or so is the appearance of epileptiform activity or seizures and sleep fragmentation and deficits in slow wave sleep. And today I'll be talking primarily about the epileptiform activity and seizures. So these mice develop aberrant synchronous epileptiform activity that you can see here. These are EEG traces from a non-transgenic animal on the left and an APP mouse on the right. These are leads in the left and right frontal, temporal, parietal, and occipital lobes. You can see normal EEG in the non-transgenic animal, but these synchronous sharp waves in the APP mouse. And you can also see occasional spontaneous non-convulsive seizures in the APP mice themselves. So what do we know about epileptiform activity in Alzheimer's disease? Well, there's a 5 to 10-fold higher seizure incidence in Alzheimer's disease compared to reference populations, and that reaches an 87-fold increase in seizure incidence in early onset familial autosomal dominant Alzheimer's disease. There have been many clinical reports of non-convulsive seizures, partial complex seizures, but these may be underestimated due to the subtle behavioral phenotype that they're associated with. It turns out that seizures are observed in very early stages of mild cognitive impairment, or MCI, and early stages of AD, beginning near the onset of cognitive decline. And recent studies from Leonard Mucke's lab showed that 24-hour EEG, MEG recordings revealed subclinical epileptiform activity in 42% of the patients that were studied. In addition, Jeff Noble's lab, together with researchers at Harvard, showed that electrodes placed in the foraminal valley revealed non-convulsive seizures, while the simultaneous scalp EEG was unremarkable. Therefore, it was previously thought that seizures were secondary to Alzheimer's disease, but now it appears that seizures might actively contribute to cognitive impairment early in the disease. Notably, anti-epileptic treatment of both patients and mice controls epileptiform activity and improves cognition. And so this leads to two major areas of research in the field. First, how does A-beta cause seizures? And second, how do seizures cause cognitive deficits? And this is a particularly interesting question for my lab because we know that the seizures are important for cognitive deficits since anti-epileptic treatments can improve cognition. However, these seizures are not frequent. They're intermittent. They can be fairly infrequent, but the cognitive deficits persist even during seizure-free periods. So what my lab has done over the last few years is to really address this, to really understand what seizures are doing to cause these long-lasting cognitive deficits. And I'm going to focus on two areas. First is epigenetic regulation of gene expression, and second is altered neurogenesis. So we've done some work and published a few papers in the last few years that have demonstrated that recurrent seizures lead to the expression of activity-dependent delta FosB, a transcription factor with a very long half-life of about 8 days in vivo. If you can see here, the related transcription factor CFOS has a very acute increase in expression, but it's short-lasting and it's gone by within a few hours. However, delta FosB takes longer to go up with activity, but it persists for long periods of time. And if you have chronic activity, then delta FosB accumulates over time in the nuclei of very active neurons. And you can see that here in the dentate gyrus of the hippocampus of a non-transgenic mouse on the left and an APP mouse here on the right. And the increase in delta FosB correlates beautifully with the amount of seizures or frequency of seizures per day. The increase in delta FosB is also associated with impairments in the Morris-Water maze, a test of learning and memory. And treatment with anti-epileptic drugs like levetiracetam decrease the expression of delta FosB in APP mice. And we know this is related to seizures because of this anti-epileptic drug treatment, and also because if we look at delta FosB in the hippocampus of a wild-type mouse that is treated with a chemoconvulsant to induce epilepsy, we see that same kind of increase in delta FosB. And what delta FosB does is to epigenetically regulate target genes. As you can see here, depicted of a random delta FosB target gene, delta FosB binds to the promoter of these genes, recruits histone deacetylase to deacetylate the gene, resulting in a closed chromatin structure and reduced gene expression. And we have found a few of the delta FosB gene targets that are very important for cognition in Alzheimer's disease, including CFOS and calbindin. And I will just go over these two very briefly. So the first is CFOS. These are studies done by a graduate student in the lab, Brian Corbett. He found that while most studies of CFOS is that acute activity-dependent expression gene, most studies in a normal non-transgenic mice see a proportion of cells that are active in the hippocampus. When you look at an APP mouse, there's very few of these active cells, suggesting that the hippocampus is not very active. However, we have electrodes in these hippocampi, and we know that these mice are having seizures. So how can this be? This is when Brian discovered delta FosB as a very good marker of chronic activity in the hippocampus. And you can see here in the very same mouse that whereas the APP mouse has less CFOS, it has much higher levels of delta FosB expression than the non-transgenic mouse. And you can see this on a mouse-by-mouse basis, that a mouse that has more delta FosB has less CFOS compared to the non-transgenic animals. And you can also see this on a cell-by-cell basis, where cells within the dentate gyrus the cells that have more delta FosB have less CFOS, and that's correlated here. And so Brian went on to demonstrate with CHIP studies, or chromatin immutable precipitation, that delta FosB binds the CFOS promoter and reduces acetylation, resulting in decreased expression. He was able to replicate all of these findings in wild-type, non-transgenic mice that were treated with canic acid, a pharmacological model of epilepsy. Moreover, if we block delta FosB signaling in APP mice, then we improved CFOS expression and improved memory. So these studies really suggested that, or demonstrated that CFOS is an important target of delta FosB in APP mice. We next went on to identify calbindin as a key target of delta FosB. These studies were done by an MD PhD student in the lab, Jason Yu. Calbindin has long been of interest to many people in both the Alzheimer's and epilepsy field, as it's been shown to be strongly decreased in expression in the hippocampi of mouse models of epilepsy and Alzheimer's disease, as well as patients with epilepsy or Alzheimer's disease. And it's been known to play a very important role in the cognitive deficits associated with both of these conditions. However, it has been unclear as to how seizures lead to decreased expression of calbindin. What Jason found was that calbindin is a novel target of delta FosB in the dentate gyrus. And you can see here on top, in non-transgenic mice that have normal levels of calbindin, they have very little, they don't have seizures, so they don't have much delta FosB expression. In an APP mouse with mild seizures, you start to see the increase in delta FosB expression and the reduction of calbindin. And in mice with severe seizures, there's a lot of delta FosB and very little calbindin expression. Jason found the same was true in a pilocarpine pharmacological model of epilepsy, where mice with mild seizures have mild increases in delta FosB, and mice with severe seizures have severe increases in delta FosB and suppression of calbindin. He also performed chromatin immunoprecipitation studies to demonstrate that delta FosB binds the calbindin promoter in both mouse models of AD as well as epilepsy. If he overexpressed delta FosB in the dentate gyrus of wild-type mice, he could suppress calbindin directly and impair memory. Moreover, when he blocked delta FosB signaling in APP mice, he could improve calbindin expression and memory, and direct overexpression of calbindin in the dentate of APP mice improves memory, demonstrating that it's a critical player in cognitive deficits in Alzheimer's disease. Jason went on to show that these findings were relevant to human populations. You can see here delta FosB and calbindin staining in control patients and mild cognitive impairment, or MCI patients, which is a prodromal form of Alzheimer's disease. The number in parentheses are mini mental status exam scores, where 30 is a perfect score. So you can see that in control patients that do very well on the mini mental status exam, they have very little delta FosB expression and beautiful calbindin expression. However, in MCI patients, the worse the mini mental status exam score, the higher the delta FosB expression and the lower the expression of calbindin, demonstrating this relationship in AD patients as well. We also worked with Dan Yoshua in the Department of Neurosurgery to obtain tissue samples of hippocampal resections that he had performed for his patients with temporal lobe epilepsy. And you can see here as well, with delta FosB across the top and calbindin across the bottom, that patients who had the most severe increase in delta FosB expression had the worst decreases in calbindin expression, and you can see that relationship quantified here. And so calbindin is an important target of delta FosB that is known to play a role in cognition and may play an important role in how seizures affect cognition in any condition with epilepsy. So these were our hypothesis-based approaches to identify targets of delta FosB. We also took a non-biased approach using CHIP sequencing to identify the entire repertoire of delta FosB targets in the hippocampus of APP mice. And Jason did this together with another graduate student, Gabe Stevens, in the lab, who has carried this work on. And just to very briefly summarize, what we found is that in APP mice, there's a much greater number of new or unique targets of delta FosB compared to those in non-transgenic mice. There's very little overlap. We found the same in mice treated with pilocarpine, the pharmacological model of epilepsy. So these new targets of delta FosB are revealed probably due to some differences in condition that's induced by the recurrent seizures. And we've gone on to look at what those targets are, and in both of the APP and PILO cases, we find that a lot of these targets are related to genes that are involved in excitability and neurotransmission, including those involved in plasticity and learning and memory, neurogenesis, cell stress and immunity, and chromatin remodeling. And so overall, the bigger picture for delta FosB is that we found that seizures greatly induce the expression of delta FosB, which epigenetically regulates target gene expression over long periods of time. It provides a mechanism by which intermittent seizures exert long-lasting effects on cognition, even during seizure-free periods. And identifying the repertoire of delta FosB targets and understanding how they may regulate various aspects of neuronal function will be critical for improving cognition and other behavioral deficits in conditions with recurrent seizures. And I just want to tell you a little bit about the other area of work that we've been investigating, which is how seizures affect neurogenesis. So neurogenesis in the dentate gyrus is important for cognition and mood, both of which are affected in Alzheimer's disease. There's been reports of increased, decreased, or no changes in neurogenesis in AD mice and humans. And recently it's been found that seizures increase neurogenesis, but chronic epilepsy is associated with reduced neurogenesis. And notably, this increased seizure incidence that we've described occurs in both AD patients and mice beginning in early stages of the disease. And this was very difficult to understand until a paper, now about 9 years ago, demonstrated that there is a population of neurostem cells in the dentate gyrus that have a finite ability to divide. And that is just demonstrated here. If you have neurostem cells in the dentate gyrus here in green, they typically divide to form amplifying neuroprogenitors and go on to mature into granule cells, or granule neurons. But what Jeannie Chin's lab found is that these neurostem cells have a limited capacity to divide. They divide only 3 to 4 times to form neuroprogenitors before they terminally differentiate into astrocytes and effectively exit the stem cell pool. And we found that in our mouse model, long before plaques form and there is plaque or pathology-related astrocytosis, in these mice, when they're very young, we see an increase in astrocytes. So we wondered, do seizures in AD aberrantly stimulate neurogenesis and prematurely deplete the neurostem cell pool? And this is a question that Annie Fu, a graduate student in the lab, worked on together with Dan Iasconi when he was an undergrad in the lab. They performed immunophenotyping of cell types so that they could look at the neurostem cells stained with nestin, or dividing neurostem cells that stained with nestin and BRDU, and they could also use double cortin as a marker to look at these immature neurons. And they started looking over many ages in our mice, starting at very early ages, because that is when we demonstrated that these mice first start developing epileptiform activity, where at 1 month of age they have epileptiform spikes, and at 2 months of age they start demonstrating overt seizures. So I'm going to summarize that data here, where when they look at these newborn neurons, our hypothesis would suggest that if you look at neurogenesis on the y-axis and time with recurrent seizures on the x-axis, that when seizures start, there's going to be an increase in neurogenesis. This will start to fall as the neurostem cells become depleted, and then fall below baseline when the stem cells are depleted and there's decreased neurogenesis. And suffice it to say, this is essentially what they found, that at very young ages, at 1 month of age in APP mice, there's no change in neurogenesis. There's an increase by 2 months of age, a decrease, and that decrease remains for the rest of the animals' lives. And when we looked at the number of total neurostem cells to see if this was associated with an accelerated depletion of the animals, of the stem cells, this is what we saw. So first, this is just neurostem cells in normal mice that decreases over time, which is similar to what Jeannie Chin's lab found many years ago. And then dividing neurostem cells also decrease with age. However, in our APP mice, we find that this depletion is greatly accelerated, such that by 2 months of age, they have very few neurostem cells left. And this was associated with an increase in division at 1 month of age. So they start dividing very early, they start depleting within a month, because it takes about a month for a cell from division to become a neuron. And you can see that here. And what we found is that this is indeed related to the seizures, because if we treat these mice with levetiracetam, an anti-seizure drug, at this point in time in their lives, We could prevent the accelerated depletion of neurostem cells. We restored normal levels of adult-born neurons throughout aging in these animals. And we improved performance on a cognitive task that relies on adult-born neurons. So overall, what we have found throughout these studies, these implications suggest that normalizing neuronal activity may help preserve the neurostem cell pool and improve hippocampal function over time. And treatments that increase self-renewal of hippocampal neurostem cells may be an important therapeutic strategy for Alzheimer's disease. And finally, these studies underscore the impact that really knowing the clinical history of a patient has on the presence or amount of adult neurogenesis that you might expect to see in those samples. And this has implications for conflicting studies in the literature regarding the presence of adult neurogenesis in humans. So in closing, we have been very interested in understanding what seizures, although infrequent and intermittent, can do in cases of neurological diseases that are associated with them, like Alzheimer's disease and epilepsy. There is the actual electrophysiological manifestation of the seizure that you can see on EEG, and that's what a lot of people typically think about. But what our studies have demonstrated is that there are effects that are much longer lasting of these seizures. For example, on activity-dependent gene expression, whose effects can last much longer and outlast the seizure itself for up to weeks in the case of delta FosB, which has a very long half-life. And finally, these seizures can induce phenomena that have much longer lasting effects on the hippocampus and cognitive function, such as this acceleration of depletion of the neurostem cell pool with long-lasting effects on hippocampal function. And so I'd just like to finish by acknowledging, I'm giving the talk, but I would like to acknowledge the people who actually did the work, which are the people in my lab. I've mentioned most of them throughout the talk, but it's really a team effort, and everybody in the lab greatly contributes to all of our work. And of course, the collaborators, some of whom I was able to talk about and some of whom I didn't have time to talk about today. And I really thank the funding sources and of course, Baylor College of Medicine for the wonderful community support and sense of camaraderie here for research. Thank you very much.